Don't forget to like, subscribe and share the videos.
The topic that we are going to discuss now is Punnett square. What is Punnett square? Why is Punnett square in news? Let us understand the details. Punnett square is named after the British geneticist, that is Reginald Punnett. What is it? It is a grid. Punnett square is a grid that is used to predict the possible genetic outcomes of offspring when two individuals with known genotypes are crossed. Two individuals are there, their genotypes are very well known. We'll understand what is genotype, phenotype and other details in upcoming slides. Two individuals are there, their genotype is very well known, and what are all the possible genetic outcomes of the offsprings of those two individuals. This can be found with the help of the Punnett square. It is like a grid. We'll understand every other details of it. So here, what is genotype? Genotype of any organism is its complete set of genetic material. So the complete set of the genetic material of a particular organism is genotype. Moreover, genotype can also be used to refer to the alleles or variants of an individual carries in a particular gene or genetic location. Okay. So even alleles and variants an individual carry in a particular gene or genetic location is also called as genotype. Okay, and it consists of a simple grid. What consists of a simple grid? This Punnett square consists of a simple grid with squares that is representing possible combination of traits inherited, that is inherited from each parent. Okay, each parent will be contributing to different traits of the offspring or the child. Okay, so what are all the possible combinations that can be there? This is got with the help of Punnett square. Okay, what else do we have to understand? We need to get into the basics of the Punnett square. First, we need to understand the concept of allele. We saw the term allele, and then we saw the term genotype, and then we must also understand the concept of phenotype and how is phenotype and genotype different. Okay, so first we'll understand what are alleles. Alleles are nothing but one of two or more alternative forms of gene that arise by mutation and are found at the same place on the chromosome. Okay, so they are alternative forms of the same gene that arise by mutation and they are found at the same location in the chromosome. So this is the chromosome and I have one allele here and another allele here. Okay, all these are alleles. So what is an allele? Allele for white flower is expressed by this color. Okay, and allele for purple flower is expressed by this color. Same gene, same location, but different variants of the same gene. Okay, chromosome, same chromosome are there. And then this particular gene alone is varying. This variation in the gene is called as allele. Okay, so the locus of flower color gene is here. So this is where the gene which is responsible for flower's color will be present. That is the locus, location. Okay, here we have for white flower and there you have for purple flower. So this is how it is varying. So this is allele. Okay. And what is phenotype? Phenotype is nothing but the set of observable characteristics of an individual that is resulting from the interaction of the genotype that we discussed with the environment. So what is genotype? Genotype is an organism's genetic information, as simple as that. Okay, an organism's genetic information is genotype. When that genetic information interacts with its environment, then that becomes phenotype because it will express itself in the form of observable characteristic. Okay, so observable traits, the physical traits are called as phenotype. Say for example, BB, that is homozygous dominant gene, is genotype here. Okay, whereas it is expressed in the form of purple flower because purple is the dominant one here. And then capital B, small b, this is heterozygous. They are not same, so heterozygous. They are same here, dominant and dominant. That is why it is homozygous dominant. This is heterozygous. One small b and another capital B, that is dominant and recessive. Both are present here. okay, it will express itself as purple again. This is the observable characteristic that we are talking about. That is phenotype. And when you see this, homozygous recessive. Homozygous because both are recessive, B, small b and small b. White is expressed because small b is for white flowers. So this is phenotype because the observable trait that we are seeing, the physical trait that we are seeing. So I hope it is clear what is genotype, what is phenotype, and what is allele. Okay. Next, we'll summarize this. So genes determine traits. We know that our genes determine our traits or characteristics like eye, skin, hair color of any organisms. And each gene in an individual consists of two alleles. One comes from mother and another from the father. So which is going to be the dominant one and which is going to be the recessive one? And based on the combination, you will be, your eye color or your skin color, hair color will be determined. Okay, this is the basic. Next is, some alleles are dominant, like we discussed, meaning they ultimately determine the expression of the trait. The dominant one will ultimately determine what trait will be exhibited. And then other alleles are recessive and are much less likely to be expressed. Okay, so if capital B is dominant and small b is recessive, if I have this combination BB, obviously this will be dominant. And even if I have this combination B and small b, capital B and small b, even here the dominant one will be expressed. Only when I have this combination, that is BB, both recessive, my recessive trait will be expressed. Okay, this is what we need to understand. Then when the dominant allele is paired with the recessive allele, that is what we discussed, the dominant allele determines the characteristic. That is the second case. When the dominant allele is paired with the recessive allele, the dominant one will be expressed. And when these traits or characteristics are visibly expressed, then they are known as phenotypes, that we discussed in the previous slide. And the genetic code behind these traits is known as genotype. As simple as that. Okay, I hope it is clear. Now let us understand what is Punnett square and how does it work and how can we construct a Punnett square. Okay, so the grid, how is the grid formed? First is we need to set up something. What is it? List the possible genetic traits of one parent along the top of the grid and the other parent along the side. So here I am giving one parent's trait and here I am giving another parent's trait. Similarly, this is another Punnett square. Here one parent's and here another parent's. The traits are given like this, and then combination has to be done. That is, fill in the squares by pairing each trait from one parent with the corresponding trait from the other parent. So this is how I will combine this. P, P, capital P, small p, capital P, small p, capital P, small p. So in this combination, if you see, 100% it will be purple because there is one dominant allele in every other combination. So if there is at least one dominant allele in a combination, that dominant trait will be expressed. Okay, but if you see here, it is capital P, small p, capital P, small p, this type of combination. Here if you see, capital P, capital P, capital P, small p, capital P, small p, and small p is also there. 2 recessive is also there. So here if you see, it is 3:1 combination. So 3 dominant trait and 1 recessive trait will be expressed. So these are all the different combinations. This is how we'll know which trait will be expressed or which type of phenotype will be expressed. Okay. Next is outcome. So each square represents a potential combination of trait that the offspring could inherit from the parents. So all these are just possibilities. So Punnett square is all about possibilities. These are all the different possibilities that can be expressed. These are all the different traits that can be expressed. Okay, I hope it is clear. Now let us understand the application of Punnett square. So Punnett squares are commonly used in biology to understand inheritance pattern, like we saw, like when you learn about the dominant and the recessive genes in school level. Next is, it is useful tool to help predict the variations and probabilities resulting from crossbreeding. So when I go for crossbreeding, all the different variations and combinations can be predicted. And then it can also be used to understand the genetic traits of the offspring in animals, okay, including humans. and researchers typically use them together with Mendelian inheritance method. Okay, so Mendelian inheritance method along with Punnett square will give you excellent results, outcomes. Okay, I hope it is clear.